I'm Ushma Neal with the Journal of Clinical Investigation. Thrilled that you've joined me for another one of our conversations with giants in medicine. I'm delighted to be joined today by physician scientist Charles Sawyers of Memorial Sloan Kettering Cancer Center. His research has defined the molecular lesions that cause cancer and used these insights to develop new drugs. Specifically, Sawyers was one of the critical members of the team that brought imatinib and dasatinib to bear on CML, chronic myelogenous leukemia, which used to be a death sentence and is now a chronic treatable disorder. Furthermore, his work has identified and gotten FDA approval for a new second-generation anti-androgen drug to bring to castration-resistant prostate cancer. And forgive me for warping this idiom, but Charles is not part of the resistance. Actually, his work is more about combating resistance, at least drug resistance in cancer cells. So thank you so much for joining me today, Charles.
Thank you, Ushma, for inviting me.
Can you start by telling me a little bit about your upbringing, your parents, your siblings?
I grew up in Nashville, Tennessee. I'm the child of two physicians. My father was a surgeon and rose through the ranks at Vanderbilt to be the chairman of surgery, so an academic, but clinical. Mm-hmm. My mother was an anesthesiologist. I had three uncles who were physicians. I had a grandfather who was a physician.
So was it a bit of a foregone conclusion that you would and your siblings would be physicians?
Well, perhaps, but I tried to resist it a little bit, and my younger brother and sister are not in medicine. My sister actually is, in that she runs the home health care agency at Vanderbilt.
So what were you like as a kid? Were you a good student? Were you an athlete? What kept you interested?
I was a very good student. I was kind of a brainy kid, good in most subjects, all subjects. Math and science I liked, but I also liked other things, and I played tennis, competitive tennis, all through high school. Not good enough to play in college, but I was, mostly academic, but reasonably good in sports.
So how did you decide then to head to Princeton to major in history?
Well, I decided to head to Princeton because I wanted to get out of Nashville. Although Nashville is a very cool town now, always was, but I needed to spread my wings. I think we all know what that is. And when I went to Princeton, I found that I really liked taking the more liberal arts classes. I wasn't dead set on going to medical school, but I thought I probably would. So my strategy was take the requirements as an in case, and explore other things. And the history department at Princeton is outstanding, and I just really enjoyed it.
So your senior thesis was something on the history of medicine in China, correct?
Correct. So I had the somewhat unique opportunity through my dad to visit China in 1980. And if those of you who are old enough will remember, that shortly after Nixon made a trip to China and sort of opened the gates. And so I was one of the early American trips, and my dad was invited as part of a medical sort of congregation. And I was just sort of the passenger, and I found it incredibly interesting and started reading a lot about Maoist doctrines, in particular barefoot doctors.
So was that a further cementing of your intrigue and desire to head to medical school?
I think so, as well as it sparked an interest in public health and the way medicine is organized and so forth, which was part of what I was actually initially interested in when I started medical school. I did not have any laboratory training or even a strong spark of wanting to pursue basic disease mechanisms at that time.
So you head to Hopkins for medical school, perhaps with the idea that you would do public health focused clinical practice. At what point did you find more of the scientific side of medicine?
In the second year of medical school, we finally start learning about disease, and we're being taught by many physicians who have laboratory-based programs, and that is when sort of the spark was lit. I mean, I can remember many different lectures where I just suddenly felt like, wow, there's a whole world that I don't know anything about that maybe I have dismissed too early. Because earlier, in the first year of medical school, at least the curriculum back then was just pure rote memorization of basic science. And most of my classmates had been biochem majors, and a significant fraction were MD, PhD, working in labs. And I just didn't have any interest in that other than, you know, getting my grade and moving on. But these physicians really inspired me. I can remember a lecture on the structure of hemoglobin and the, you know, understanding of the sickle cell mutations and so forth, and just the ability to reduce, you know, a complex disease down to a point mutation in one gene fascinated me. And you know, this is the this was early 80s. So molecular biology was a discipline that was just growing, and it hadn't been applied to disease in any real way. And I just thought, wow, this could be something really important, and maybe I should, you know, pay attention.
Did you get any research experience during medical school?
I did. I was able to forego many of the electives of the fourth year and spend a year in a lab. That lab was in pharmacology for various reasons that were good at the time, and gave me some exposure to experimental design and so forth. But by doing that and having the freedom that that kind of life gives you, and hang out with other adjacent labs, I met, you know, a whole new community of postdoctoral fellows and graduate students and so forth. And just, you know, probably the most valuable part of that was knowing about that career path and talking science with a lot of young people who are, you know, stimulating, working on different topics, as well as the faculty mentors of all of these labs.
But at the same time, you weren't willing to give up the clinical side of things. So you decided to do a residency in internal medicine. So you started out in the southern Midwest, then spent a fair bit of time on the East Coast, and you decided to go west.
I went to California, UC San Francisco, which I ranked first and was lucky enough to match. My other options were Boston or Hopkins, or I guess I could have gone back to Vanderbilt. But I went to UCSF because my cousin, who had also gone to Hopkins, he's about 5 years older, was a rheumatology fellow and working in a basic immunology lab, and he said, you got to come out here, it's unbelievable. And I also was avidly reading everything about, you know, molecular medicine, and there was at that time an incredible community of physicians who are MD scientists, Harold Varmus and Mike Bishop, to name just two, as well as others. And I thought, you know, I think I just need to go there and just sort of figure it out. And I'm really glad I did. It was a very transformative experience for me.
So you met Joel Ernst, who's now in New York City at NYU, but at the time was at UCSF, and he gave you a little bit of a scientific push then.
Yeah, so Joel was in some ways sort of like me, but a few years ahead. Hadn't really been on a physician-scientist track, infectious disease, and was my attending when I was a resident, probably an intern and a resident. I got to know him and we just completely clicked. And sort of like the medical school curriculum, I was able to parlay roughly 6 months of the third year of residency into a research experience in his lab. And you know, as the physicians know who are watching, you have, you know, obligations as a resident to cover various services and so forth. So the logistics of working that out was a trade where I agreed to cover the medical ICU at night at San Francisco General Hospital while I worked at, you know, at Joel's lab during the day on a pretty grueling night schedule, but it was totally worth it to me because I got to have, you know, sort of my cake and eat it too.
So was his lab at that point still focused on infectious disease?
It was, but it was cloning genes involved in white blood cell, phagocytosis of bacteria and so forth. And so that, although, I found that interesting, the sort of the fun part for both of us was we wanted to clone the cDNA for a certain gene, and we had to do it the old way, which was we had a little bit of protein purified, got some peptide sequence, and based on that made oligonucleotides to screen a cDNA library and plaque lifts from phage libraries and so forth. It was the way genes were cloned back in the early days.
So you entered medical school wanting to do public health. You did an internal medicine residency and you worked in a lab where it might not have been infectious diseases, but that was maybe the big picture. At what point did you turn towards hematology oncology?
I think I knew even back in medical school that cancer fascinated me as a scientific problem for, I think, pretty obvious reasons. Cancer is yourself, and genes are going awry. Oncogenes were now being described. The fundamental Bishop Varmus thesis was just starting to gather momentum, and I just knew that something big was here. But I hadn't been exposed to the clinical side. It's just medical students at Hopkins don't see many cancer patients. Maybe that's changed now. But when you're an intern and resident, you do, and I just, I knew it was what I wanted to do clinically when I was on the leukemia service at Moffitt Hospital. So just again to sort of frame the timing, this is when allogeneic bone marrow transplantation was relatively new. Not very many centers had the infrastructure to do it, and UCSF was one. And interestingly, chronic myeloid leukemia was a disease in which it was really starting to show exciting outcome data. So that also sparked an interest in learning everything I could about chronic myeloid leukemia.
Did you ever have a patient that either swayed you one way or another or solidified that decision?
I do remember vividly one patient who had chronic myeloid leukemia in the chronic phase, was my age, married, was a fireman, and again, it's the early days of transplantation, and today, obviously he would be treated differently, but it made sense for this young man to have an allogeneic transplant, and he unfortunately died of graft versus host disease. So that was impactful for a lot of reasons. One, essentially a person who is otherwise healthy, having to make this important decision. The second, realizing that this is an unfair therapy to have to make someone choose to undergo. We have to do better, and that's when I started reading about the Philadelphia chromosome.
So that brings us to the next phase of your career, which is your Hem/Onc fellowship, which you took at UCLA. Did you seek out Owen Whitty's lab to go and work there specifically because of these experiences? I
did, and I was interested in the disease. I was reading all of the early papers from Noel and Hungerford on the description of the chromosome at University of Pennsylvania, and then Janet Rowley's discovery through cytogenetic banding that it was a reciprocal translocation, and then the race to try to clone the translocation. Joel, to his credit, pointed me to a paper that had just come out from Owen Whitty's lab at who had just moved from David Baltimore's group, where he was a postdoc, to start his group at UCLA. And he had cloned the translocation and had the first cDNA. And I thought, and he was a physician, hadn't done clinical training, but I thought, I think this is a sign. I mean, I wanted to stay in California as well, although, from San Francisco, moving to LA, I had to, swallow that pill. But it was the best decision I made. And so I actually contacted him, as a third-year resident, working in this lab. And again, for those of you who are young, I had to actually get out a typewriter and write a letter and put a stamp on the envelope and mail it. And he wrote me back and said, call me, and then we talked, and I went down for a visit. And so I was going to go there to do a postdoc. And then I quickly put in a fellowship application sort of a little bit after the deadline, and a spot opened up. So it ended up being, working out, obviously, perfectly well. Yeah, I think that, Owen's lab was a special place for many reasons. It was a big lab. He was incredibly successful at that point, and so one of the top draws for the best postdocs in the UCLA biomedical community. He had a reputation of being a little rough around the edges, and not many clinical people, felt that they could sort of hold their own with him. I don't know why, but he seemed to take an interest in me. Maybe because my path was a little different and I was clearly determined. And we were able to work it out so that I actually started in his lab before I started my clinical fellowship. And the timing was very serendipitous with the recent discovery of PCR. And this is how far back, this is how old I am. So the question was, could you detect the BCR-ABL transcript by PCR? Obviously you could, but that was a pretty novel idea back then. And then could we apply it to a real life clinical scenario? So I learned the technology, got it working on all the sort of laboratory reagents, and then to Owen's credit, he let me sort of work through one of the technicians to keep the project going when I then started in the clinic. But of course, I think he really had played this out in his head pretty well, because he knew I would now have all the connections and access to the samples from patients that would really make this story into something important. So we actually ended up having a nice paper showing we could track low levels of transcripts in patients who had had an allogeneic transplant. The prognostic impact was not clear at that time, but now it's a standard test to follow patients.
So at what point did you and Brian Drucker and Nick Leiden meet?
Owen, early on in my postdoc, trusted me enough to represent the lab or along with one or two other postdocs. So I started going to that when I was still a postdoc. And Brian was in Tom Roberts' group, and then also working with Jim Griffin from the Farber. And so we were basically, equivalent stages roughly, representing, different groups interested in CML. That's when we first met, sort of clicked because of our backgrounds. Brian also had a non-traditional, path, straight MD, no real prior research experience until he was a postdoc. So, birds of a feather, I guess. And stayed close when we each got our jobs. Brian moved to Oregon Health Sciences as his first job, where he still is and is now the Cancer Center director.
So at what point did you two start putting your heads together around a potential molecular target?
Brian invited me as a guest speaker when he was an assistant professor. I can't remember the exact year, but it was in the early 90s. And showed me some data with this compound that Nick Leiden and colleagues had sent him from what was then called Ciba-Geigy Pharmaceuticals against a few cell lines. And it looked pretty interesting, but it's very hard at that stage, first of all, of your career, as well as that stage in the development of a compound, to know, is that something you should put a whole big effort behind or not. But he asked me to sort of help him kind of champion the idea, look at it myself, and see if we could convince this Swiss pharmaceutical company, who didn't really know much about this program, to move it along. That then began a long, extremely exciting, fruitful collaboration with many bumps along the way in terms of Ciba-Geigy merging with Sandoz to become Novartis, and convincing clinical development teams that it was worth spending some money on this. But obviously, you look back, it was a spectacular success.
So talk to me a little bit about what it felt like. So you, together with other colleagues, were the first ones to write the protocols and run the first clinical trials on imatinib that later became known as Gleevec. What was it like as you were getting some of the data back on the dose escalation trials and realizing that this could have some legs?
It was a little bit pins and needles, and not wanting to get too ahead of ourselves in terms of level of excitement. But like any phase 1 trial in cancer, it was what's called the classic 3+3 dose escalation, where 3 patients received the initial starting dose. Nothing happened. It was a very low dose. There was actually a hint at either first or second dose level that one patient's white blood cell count seemed to get better, but it was just random. But at a dose, the standard dose, I believe, is still 400. But at the dose level of 300, all 3 patients had a decline in their white blood cell count. So we knew that was likely to be real, and it, of course, repeated. And then we knew there was a drug, that it had activity. But the question at that point was, is it going to be any better than just taking oral chemotherapy? Because we had the ability then to control the white blood count of CML patients. So the real test of true benefit is to test the bone marrow for the presence of the Philadelphia chromosome. And those results started coming in after patients had been on active doses at 6 months. And I was lucky enough to have the first patient who had a complete cytogenetic remission. And I remember this because I was in my office on a Saturday morning, sort of getting ready later that day to fly to a meeting in Europe where Brian and I were going to discuss the first clinical data in a small group meeting that included people from Novartis. And that back then we would get the results from the cytogenetic analysis on the fax machine. And the fax machine starts whirring in my office, and there's this report, 0 out of 20, and I could not believe it. I called the lab to make sure, and then I tried to call Brian, and he had already left for the airport, so I didn't have anyone to call, so I actually called the patient who lived in Santa Barbara, still does, and then I went to the airport. And
did you have some champagne on the flight?
I had some champagne with Brian and colleagues the next day. But one is only one, and it took a few more months for the rest of them to come in, and it was amazing. I'd say, another really important part of this that is completely unforgettable to me is the decision that we made early on after we were seeing the clinical benefit in the patients in chronic phase to open it up to patients with blast crisis. So, this is the phase of the disease that is lethal, within, months. Usually the median survival was 3 to 6 months at that time. And the cytogenetic landscape of those patients is complete chaos. So the idea that BCR-ABL was a driver of the entire disease seemed ludicrous. But we felt ethically we had to at least take a look. And it was incredible how many patients who were near death's door went into a complete remission within a matter of weeks of taking this pill. That probably was the most sort of revealing moment to me, that cancer, even though it's genetically complex, has an Achilles heel. And I think that's when I sort of had a kind of a point in my mind where, at my age, with what I know, this is what I have to do. I have to figure this out, and sort of galvanize the entire cancer research community, that this isn't just CML, which isn't really cancer, this is real cancer.
But then you see some of your patients starting to relapse.
Not only are they relapsing, they're relapsing very quickly. So I felt like we had to understand resistance as quickly as possible. Otherwise, this is all going to go by the wayside. And although I hadn't carefully studied all the papers in resistance to HIV, early antiretroviral therapy, I was aware enough of the euphoria of success and sort of the agony of defeat. And then the power of understanding an acquired resistance mechanism at helping derive combination therapies. So I was like, I don't know if that's going to be the case here, but let's figure it out. And I was lucky enough to have the right people in my lab to really push this question. So, everyone had their own project in my lab that was, focused on various aspects of BCR-ABL signaling. But as soon as I saw the first couple of patients relapse, I was like, I had to, sort of catalyze the group to say, we have to make this the highest priority project. And let's start brainstorming on how to do it. And the people who made that happen are Mercedes Gore and Neil Shaw, who were critical in discovering that resistance was caused by new mutations in the BCR-ABL kinase domain that confer resistance.
How did you then start to dip a toe in the water of structural biology?
The first mutation, we got it right, but we oversimplified. We were a little naive with the first paper. We sequenced 13 patients, and 11 of the 13 all had the same mutation. So I was like, oh my God, there's only one resistance mechanism. So that meant we just need a second drug. Structure actually, and what was also compelling about that is the mutation was in what is now famously known as the gatekeeper residue, which is shared, is present in every kinase. And a lot of the ATP competitive inhibitors work by binding to that gatekeeper mutation, that gatekeeper residue. So it's a simple way to get resistance. Mm-hmm. But with more analysis of more patients, et cetera, it quickly became clear that there were numerous mutations that could all confer resistance. I think that the count is up to 50 or so. So that's when really getting into the structural biology was important, because we can't develop 50 new drugs to handle each mutation. And the sort of good timing, again, was John Kurian's lab had solved the first ABL crystal structure. He had moved from Rockefeller to Berkeley. We had met through our first discovery. I'd called him and shared data, and we had met in person. He said, why don't we just have people from our groups meet each other periodically and review the data? So we went up to Berkeley one trip with a small number of people from my group and mapped out all the mutations using the software that they have to view structures. And it became clear that what these mutations, for the most part, were doing was changing the conformational flexibility of the kinase. So it gets a little into the details, but Gleevec is very special in how it binds ABL. ABL has to be in a conformation that's completely inactive, which it flips into in and out, for a microsecond normally. And what a lot of the new mutations do are prevent it from being so flexible. So we just needed to have, so the idea was find an inhibitor that can tolerate these other conformations, and dasatinib is that drug. And now there are 3 other approved ABL kinase inhibitors, all iterating on all of this work. So, so CML is a fantastic story for patients, for science, for molecular understanding of disease.
But you also had turned your attention to prostate cancer. So at the same time as all of these things are coming out around CML, you're developing new models for prostate cancer and thinking about it that way. So Two questions. First off, why prostate? Why not lung cancer? Why not ovarian cancer, something else? And then tell me a little bit about that journey.
The prostate decision was influenced by a number of variables. One was there was a urologist lab right next to my lab. This person needed some help, and I was a young, assistant professor, and I was like, okay, well, there's a little time, so I'll help. And as I learned more about it, I got pretty interested in the idea of, why does hormone therapy work, and why does it stop working? But there were these other diseases, and so I actually made a conscious effort to go to a few meetings of other disease areas and try to not just be a fly on the wall, but sort of gauge the status of the questions the big questions in the fields. And prostate attracted me. So at the time, this stage of disease was called androgen-independent prostate cancer or hormone-refractory prostate cancer. And the reagents in which this question was being addressed in the laboratory was largely 3 different cell lines, 2 of which no longer expressed the androgen receptor, all 3 of which were independent of, well, actually the one that was androgen receptor positive responded a bit to androgen ablation therapy. So it seems obvious in retrospect that the receptor must still be working in what's now called castration-resistant disease, but it was not the main idea, and no one was trying to develop better androgen therapy drugs, and neither were we. So we took a completely unbiased approach. Actually, the very first paper we published in prostate cancer was to develop a whole new set of models, the equivalent of what we now call patient-derived xenografts or PDX models, which we would grow in male mice, and then we would castrate the mouse, and the tumor would regress, and then it would regrow. So these matched pairs, these isogenic pairs, were the substrate for what was, again, a new technology, gene expression arrays, now RNA-seq, but long and short of it is a project that was supposed to discover a multitude of different resistance mechanisms, all pointed to the androgen receptor at the end. And I remember the lab meeting where this was discussed, and I was like, did we discover something important or not? And it was clear that we had to do the functional studies. What the data said, the androgen receptor is more highly expressed every time the tumors develop castration resistance. So again, technology came to the rescue. This was right when RNA interference was now able to be applied to mammalian cells using stable hairpins. Very early days of that. So a brilliant MD PhD student in my lab named Derek Welsby developed stable hairpins targeting the receptor and showed that we could flip the castration-resistant tumors back to castration-sensitive if we just knocked down the receptor. And conversely, we could overexpress and gain a function. So everything matched up. That was an important paper, defining paper for our group in the field, and led us to then say, there has to be another drug. I mean, we know the target. We just need to hit it better and screen for compounds in the right context. At the time we started this, so I now had a little bit of street cred in drug development, having done Gleevec and Sprycel, but I really didn't know how to make a drug, and I never thought we would, but we wanted to get to some kind of proof of concept with a tool compound that could, point the pharmaceutical industry towards this target. And what was fascinating, and I knew enough people to talk to, pharma, this was such a yesterday idea, and everyone had moved on to kinase inhibitors. And so at one point, Mike and I said, we just got to do it ourselves. And the key mover chemically here is Mike and a postdoctoral fellow named Samadi Ook. And we had a really well-designed cell-based screen where we knew exactly the kind of activity profile that a compound that could solve this had to have. Because the drug that existed at the time, called bicalutamide, could be overcome by overexpressing the androgen receptor. So it seems simple now, but we just recreated that context, made a really high throughput screening platform, and out came enzalutamide.
Do you ever stop and reflect and have a real sense of gratification at the sheer number of lives you've extended?
To me, the more gratifying thing is when just out of nowhere, someone who knows that I was involved comes up to me and says, my dad is on enzalutamide, thank you. Or patients on Gleevec come up to me and say, I had CML and I'm now in remission, or I had gastrointestinal stromal tumor, or it's the one-on-one ones that really hit home.
If you couldn't be a physician or a scientist, what do you think could have captivated you all these years?
I would not have gone into business or law, I know that, but I feel like I'm an academic at heart. I would have probably been a journal editor or something like that. I love reading, I love writing, and I like just talking about ideas. So.
Professional tennis player, perhaps?
No, that's not in the cards.
All right, well, thank you so much, Charles, for joining me for this. It was really great to hear a little bit more about how it felt to go through all of that.
Thank you for inviting me to join you.